Hello students, welcome to EPG Pathshala. I am Meenal Dhal from Department of Anthropology, University of Delhi. Today we will start with the module Haemoglobinopathies from the paper Human Population Genetics. The learning objectives of this module are: you shall be able to learn the history of hemoglobin research. You shall be able to understand the hemoglobin molecule and the globin genes that play a very important role in our body. And you would also know the meaning of haemoglobinopathies, how to identify it, and what are the endemic and sporadic hemoglobin variants. So let's start with the introduction of hemoglobin. Hemoglobin is the protein molecule in red blood cells that carries the oxygen from lungs to the body's tissue and return carbon dioxide from the tissues back to the lungs. The normal function and total amount of hemoglobin depend on its adequate synthesis and precise structure. When these conditions are not met, the red cells of the carrier contains either a decreased amount of hemoglobin or an hemoglobin variant that may have abnormal properties. These conditions are collectively described as haemoglobinopathies. History of hemoglobin research. Human hemoglobin started with the investigations on sickle cell anemia, a hereditary disease. Herrick observed a sickle-shaped abnormality of red cell structure in an anemic black student in 1910. Taliaferro and Huck recognized that the condition is hereditary. Neal and Beat had shown that patients with sickle cell anemia are homozygous for a gene that is in the heterozygous state, causing much milder condition, sickle cell trait, which was found in about 8% of the American black population. The decisive step in the biochemical genetic analysis of this disease was carried out by Pauling et al. In 1949 in a paper with the programmatic title Sickle Cell Anemia, a Molecular Disease. The author examined the hemoglobin of patients with sickle cell trait and sickle cell anemia, comparing them with the hemoglobin of normal individuals. In accordance with the state of methodology for protein analysis at that time, these investigations were performed using the tessellous zone electrophoresis. The results conclude with a significant difference exists between the electrophoresis mobility of hemoglobin derived from erythrocytes of normal individual and from those of sickle cell anemic individuals. The gene in sickle cell anemia is in homozygous state, whereas the carriers of the sickle cell trait are heterozygous. This reveals that a change produced in a protein molecule by an allelic change in the single gene involved in synthesis. In 1956, Ingram, working in Cambridge in the laboratory where Perutz was pursuing his crystallographic work, Sanger had shown the amino acid sequence of insulin, and Crick and Boston had demonstrated the DNA model. The fingerprinting method of protein analysis had revealed that sickle cell hemoglobin was identical with the normal molecules in all peptides except one differing peptide where glutamic acid was replaced by valine. Meanwhile, after new methods of electrophoresis had replaced the cumbersome tessellous electrophoresis, many other hemoglobin variants were also discovered. At present, over 400 of such variants are known. Further, steps of great importance were the establishment and elucidation of the full amino acid sequence of the hemoglobin chains by Bronzier et al. Given by 1961, and of the three-dimensional structure of hemoglobin. The isolation of the hemoglobin mRNA led to new insights into gene structure and function and opened up new paths to the understanding of gene action. Molecular work on the hemoglobin has proceeded at a rapid rate The full DNA sequence of the various hemoglobin genes and their flanking sequences is now known, and the hemoglobin genes are better understood than any other mammalian genes. Mutations affecting the hemoglobin, particularly the thalassemias, have been elucidated and are models for the understanding of gene action at the molecular level. In hemoglobin molecules, there are different types of hemoglobins in human. Basically, four types of hemoglobin which appear in human lifetime. The first one is the embryonic hemoglobin, that is HB Gore 1, 2 and hemoglobin Portland, which are detectable from the 3rd to 10th week of gestation period. The second type is the fetal hemoglobin, that is abbreviated as HbF, the major hemoglobin at the time of birth. The third one is the adult hemoglobin, abbreviated as HbA, and the fourth one is the minor adult hemoglobin, called it as HbA2. During the first year of the life, HbF is replaced by HbA and HbA2, where a normal adult man contains 98% of hemoglobin A, that is HbA, and 2% of HbA2. Let's talk about the hemoglobin structure. The hemoglobin molecule consists of two pairs of identical amino acid chains that interweave in space, forming an ellipsoidal tetrahedron. Its molecular weight is approximately 64,000 Da. There are several types of chain. Each has a precise amino acid sequence that is fully conserved from generation to generation. The various chains display a marked sequence similarity. They have similar amino acids in several positions across their sequence, implying a common ancestral form. The type of hemoglobin molecule is determined by its constituent chains. Adult hemoglobin is composed of two alpha peptide chains, each consisting of 141 amino acids, and two beta peptide chains, each consisting of 146 amino acids, whereas in minor adult hemoglobin, two beta peptide chains replaced by delta chains, and the fetal hemoglobin has two alpha peptide chains instead of beta peptide chains. Adult hemoglobin is composed of two alpha peptide chains, each consisting of 141 amino acids, and two beta peptide chains, each consisting of 146 amino acids, whereas in minor adult hemoglobin, two beta peptide chains replaced by delta chains, and the fetal hemoglobin has two alpha peptide chains instead of beta peptide chains. Here you can see the figure where chromosomes 16 and 11 are there. The globin gene clusters and the respective chains were also shown in the figure. The genes are arranged from 5 to 3 dash according to the timing of their activation. The green sections represent translated sequence called as exons, and the red section denote intervening sequences called introns. Blue sections correspond to the untranslated 5 to 3 dash sequence flaking the globin genes. Combination of the chain produced result in the various types of hemoglobins, which we will discuss later. Hemoglobin functions. The structure of the hemoglobin is complicated, which makes the molecule to have its accurate and long-lasting function. For example, each heme acquires an oxygen molecule which is inserted between the respective distal histidine and iron when the red cells pass through the lung capillaries, where the oxygen tension is 60 mmHg, that is 60 millimeter mercury. The four hemes bind and release oxygen sequentially. Moreover, binding of oxygen by one heme facilitates the binding or release of oxygen by the next heme. This allostatic function is already discussed in the figure by the sigmoidal shape of the oxygen equilibrium curve, which displays a horizontal part when the PO2 varies around 60 to 80 mmHg, reflecting the adequate saturation that is close to 100% at all potential fluctuations of the oxygen pressure in the air, the airways and the alveoli. A steep oblique part of PO2 values decreasing from 60 to 20 mmHg, thus ensuring a satisfactory gradual release of oxygen when red cells pass through the tissue. And lastly, the final horizontal part at partial oxygen pressures lower than 20 mmHg. Equally important is the fact that on binding oxygen, each chain undergoes a redistribution of the low energy bonds and slightly change its shape. As a result, the whole hemoglobin tetramer acquires an R, that is relax conformation on oxygenation, and squeezes into a T conformation on giving up the oxygen. The later change and release of oxygen is facilitated by the introduction of a 2,3 diphosphoglycerate molecule in the central cavity. This 2,3 diphosphoglycerate molecule is a product of Rappaport-Lubering shunt of the glycolytic Embedden-Myogloff pathway, and its concentration is adjusted to a certain degree depending on tissue needs for oxygen. Thalassaemia represents a group of hereditary disorders of hemoglobin synthesis which varying severity. The production of normal hemoglobin, that is HbA, is inhibited due to anomaly in the orderly synthesis of one or the other peptide chains of the hemoglobin molecule. They differ, however, from the other disorders of hemoglobin formation in that no abnormal hemoglobin chains are formed. Rather, the rate of adult hemoglobin formation is diminished, and as a consequence, various combinations of normal peptides chain may exist in abnormal quantity. The thalassemia results from inherited defects in the synthesis of the globin chains of hemoglobin, and humans have four hemoglobins at different stages of development. All the four different hemoglobins are tetramers of two pairs of unlike globin chains. Every individual globin chain has a heme moiety attached to it, to which oxygen is bound. With the advance in the knowledge of the structure of the hemoglobin molecule, it is now possible to distinguish a great majority of thalassemias. The two commonly found thalassemia are beta thalassemia, in which beta chain synthesis is reduced, and alpha thalassemia, where alpha chain synthesis is affected. Both alpha and beta thalassemias may exist either in the heterozygous state, when the gene for thalassemia is inherited from either father or mother, or in the homozygous state, when the gene is inherited from both the parents. Haemoglobinopathies. We have discussed or emphasized that oxygenation of red cell is performed by a very complex mechanism, where the main important points are: first, the adequate production of red cell; second, a satisfactory complement of the latter with hemoglobin molecules; and thirdly, a very precise structure of the respective globin chains. The conditions where these three important points are not met, then there is an abnormality in the hemoglobin which is known as haemoglobinopathies. Thalassemia is termed when the condition is completely or partially inhibit the rate of synthesis, whereas variants are termed when there is a change in the structure of the primary globin chain. In hemoglobin variants, each nucleotide across alpha and beta gene sequences are expected to have a chance of mutation. However, several mutations, especially those of the third nucleotide of the respective triplet, do not result in amino acid replacement. because the change still codes for the same amino acid. In fact, of the 2600 possible mutations of the alpha and beta genes, only 1690 are expected to result in an amino acid substitution, and of these, only about one third have so far been identified, approximately 200 alpha chain and more than 300 beta chains variants. This number is continuously increasing. Hemoglobin variants that are used earlier to characterize with the capital has been shifted today to the correct annotation, which gives the type of chain, the number of the amino acid in the helical or non-helical sequence, and the abbreviated amino acid change. For example, hemoglobin S is referred to as beta 6 A6 glutamic acid valine. Alpha thalassemia. A normal human receive alpha gene from each parents. So genetically it's very complicated. There are two main classes of alpha thalassemia. One is alpha O thalassemia where both alpha genes are deleted. That is, all or part of the gene is missing. The homozygous state is written as dash and the heterozygous state is written as dash oblique alpha. The other one is alpha plus thalassemia where only one of the alpha gene is lost. The homozygous and heterozygous states are designated as minus alpha slash alpha alpha and minus alpha slash minus alpha respectively. Sometimes alpha plus thalassemia results from a mutation that inactivates alpha globin genes rather than deleting it. In this case, the heterozygous state is written as alpha T alpha slash alpha alpha. 180 or more different mutations of the beta globin genes are found in patients with beta thalassemia. They may affect gene function at any level between transcriptions, processing of the primary messenger, ribonucleic acid transcript, translation, or post-translational stability of the gene product. Rarely, beta thalassemia, like alpha thalassemia, may result from a partial or complete deletion of the beta globin genes. Some of these mutations cause an absence of beta chain production, and the resulting disease is called beta O thalassemia, whereas others result in a reduced output of beta chain, beta plus thalassemia. Some of the later forms are extremely mild and may not identifiable in carriers. Most heterozygotes for beta thalassemia have very mild anemia and a raised level of HbA2. The hallmark of all the thalassemias is imbalanced globin chain production. In the beta thalassemias, this results in an excess of a chain which precipitate in the red cell precursors, leading to their damage in the bone marrow and shortening the survival of their progeny in the peripheral blood. The pathology of alpha thalassemia is different. In the face of defective alpha chain production, excess gamma chains produced in fetal life form gamma 4 molecules, while in adult, excess beta chain form beta 4 molecules. These homotetramers are called HBS BARTs, that is gamma 4, and H, that is beta 4 respectively. They do not give up oxygen affinity of the homotetramers, leads to reduced oxygen delivery to the tissues. Now, co-inheritance of thalassemia with hemoglobin variants. Most of the hemoglobin variants are rare, except there are three which are found very common in where they are hemoglobin S, hemoglobin C, and hemoglobin E. Hence, it is common for a person with beta thalassemia to co-inherit a gene for one of these variants. The compound heterozygous state for beta thalassemia and the sickle cell gene, sickle cell beta thalassemia results in a clinical picture very like sickle cell anemia. On the other hand, the inheritance of beta thalassemia together with hemoglobin E, a hemoglobin variant that is produced at a reduced rate and hence is associated with a mild beta thalassemia phenotype, produces a severe form of thalassemia which is usually but not always transfusion dependent. Hemoglobin E beta thalassemia is one of the commonest form of severe thalassemia and is assuming a major public health problem in parts of India and further east, particularly in Thailand and Indonesia. Endemic hemoglobin variants, the first one is hemoglobin S, that is beta 6A3 glutamic acid to valine. This is the most commonly found hemoglobin variant, mostly found among the black people but also found among the Mediterranean Sea and in India. Sickle cell hemoglobin, abbreviated as HbS, is a molecular abnormality of beta globin chain with the substitution of glutamic acid by valine at 6th codon position. This particular mutation grossly affects the solubility and crystallization of this hemoglobin under conditions of hypoxia. Hypoxia is the condition where there is a lack of oxygen. Sickle cell disease is an autosomal hereditary disorder where the red cells become a sickle shape, and this disease resulting from homozygous condition is a hemolytic anemia. The disease is characterized by enlarged spleen, painful crisis, organ damage, impaired mental function, increased susceptibility to infection, and ultimately early death under certain conditions. The second hemoglobin variant is hemoglobin C, where beta 6A3 glutamic acid to lysine is the conversion is to lysine. This hemoglobin variant is also common among the black people in West Central Africa. This variant resulting from homozygosity will lead to decreased solubility, target shaped red cells and hemolytic anemia, as it is also in HbS or hemoglobin S. The third type or variant of endemic hemoglobin is hemoglobin E, where beta 26 B8 glutamic acid to lysine. This variant is mostly found in the Far East, where the frequency of heterozygotes range from 10% to 25%, mainly Thai and Chinese but not Japanese or Polynesian populations. The variant chain is associated with mild instability and a slightly reduced rate of synthesis through the abnormal additional activation of a cryptic splicing site and wasting of some beta E mRNA. As a result, homozygosity for the variant causes mild hemolytic anemia, but it becomes very severe with compound beta E by beta thalassemia heterozygotes. The fourth one is hemoglobin D Punjab. It is the beta 121 GH4 glutamic acid to glutamine. This hemoglobin D variant is found in India and was first reported from Pune in a Sikh soldier. Later, further investigations in Punjab showed further incidence of HBD in Sikh and Punjabis, and also some parts of India such as Uttar Pradesh, West Bengal, Assam, Gujarat, Mumbai, Goa, Tamil Nadu, Kolkata or Kerala. It is not usually associated with any clinical manifestations. The fifth type or variant is hemoglobin O Arab, that is beta 121 GH4 glutamic acid to lysine. This hemoglobin variant is found among the Pomaks, probably representing an ancient Thracian tribe in Greece and Bulgaria. Its homozygous state is associated with mild hemolytic anemia, which is aggravated in compound BO by B thalassemia heterozygotes. Another type of variant is sporadic hemoglobin variant. The most common examples of sporadic hemoglobin variants are hemoglobin with increased oxygen affinity, hemoglobin with decreased oxygen affinity, hemoglobin M, unstable hemoglobin, hemoglobin with more than one mutation in one chain, hybrid hemoglobin, thalassemic haemoglobinopathies, hemoglobin with elongated chain, and hemoglobin with shortened chains. Now, what are the identifications of hemoglobin variants? First, we'll discuss about hematological parameters. The reason why majority of the hemoglobin variant display normal in red cell indices is that the amount of the variant hemoglobin synthesized in the red cells of a heterozygote is expected to be equal with the hemoglobin A or HbA. Many patients with sickle cell anemia are in reasonably good health most of the time and achieving a steady state level of fitness. The importance of early recognition and subsequent clinical and hematological assessment of the disease are greatly facilitated by familiarity with the patient's steady state. A patient with sickle cell anemia is said to be in steady state when there is absence of infection, acute complicating factors, or acute clinical symptoms, or crisis for at least 3 months. The levels of total hemoglobin have no way to change except in some conditions such as mutations that alter the oxygen affinity of the variant and result in anemia or polycythemia. Of course, increased numbers of reticulocytes, lactate dehydrogenase, and indirect bilirubin levels indicate hemolysis, while the detection of Heinz bodies in red cells points to a promptly precipitating hemoglobin variant. In other cases, the presence of the variant is indicated by other specific properties such as abnormal polymerization and low solubility, which produce distinct alterations in red cell morphology. Now, there are various biochemical findings also. The main approach of identifying the hemoglobin variants lies in its demonstration and quantification by biochemical studies such as electrophoresis, chromatography, spectral analysis, oxygen affinity, biosynthesis, solubility and thermal stability, peptide analysis, and DNA or molecular studies are used in the identification of hemoglobin variants. Now let us summarize what we read in this module. Broadly, the term haemoglobinopathies circumscribe a large group of inherited disorders involving the amount or structure of hemoglobin molecules which are contained in the red blood cells of human and other higher species. Precisely, the term applies mainly to the structural abnormal globin chains which results in variant hemoglobins with mild or severe consequences for the carrier. The hemoglobinopathies may occur in high frequencies with a varying clinical and social impact in various populations over the globe, or they occur as a rare events, some of which represent a unique experiments of nature and give us clues to the understanding of hemoglobin function. Thank you.